This Week in Virology, the podcast about viruses, the kind that make you sick.
Can anybody hear me?
Oh, yeah.
Yes.
Oh, it's like a disembodied voice. Hello, Chicago.
Yay, yell, come on. Yay!
I couldn't hear that from my remote location.
I was wondering
folks.
if you could. Yeah, they yelled. It was a little lame, but it was okay.
Well, we're virologists, so what do you expect?
Well, the problem is you're DNA virologists, right?
Wait a minute.
We can get some feuds going here.
One more try. Hang on. One more try. Hello, Chicago. That was much better.
Yeah, a little goading always helps. Okay, from Microbe TV, this is TWiV, This Week in Virology, a special episode recorded at the 2024 International HBV meeting in Chicago. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Chicago, Rich Condit.
Hi there, Vincent.
How's the weather out there, Rich?
There was applause there.
I got that,
You got that? Okay. It's great to be here. It's been a wonderful meeting.
And the weather, Rich?
Oh, I'm sorry. I did look it up. I haven't been outside in 3 days. It's 78 degrees and sunny. It's gorgeous, and it's been that way the whole time.
Here on the East Coast, it's 27 and sunny. Quite a nice day, although, as you can see, I'm inside. We have an episode for you from this meeting. Obviously, we're going to talk with two HBV scientists. And joining us today from University College London, Mala Maini. Welcome to TWiV.
Hi. Hi there, Vincent.
Hi, Mala. How are you doing today?
Great, thank you.
Good. Thanks for joining us. And from Washington University in St. Louis, John Tavis. Welcome to TWiV.
Thank you very much, but I'm from St. Louis University at St. Louis, not Washington University.
St. Louis University, which is in
St. Louis.
St. Louis, right? That makes it easy. I knew I'd get one of them wrong. All right, thank you, and sorry about that. We'd like to start by learning a little bit about your training. Mala, why don't you start off? Tell us how you got into science.
Well, actually, I started out for many years in medicine. So I had my father is also a medic and an immunologist, actually, like me. So I grew up seeing patients on the wards at Christmas and playing with test tubes. And then actually a real formative push for me to go into medicine was one of my brothers died when I was 15, and that sort of made my path clear that I wanted to become a medic. And for many, many years, I just practiced as a doctor and avoided anything very scientific. And then a chance encounter over a coffee when I was about to become a consultant physician persuaded me to go into the immunology lab and start trying out research. And I started a PhD. Actually spent the first year wanting to run away and go back to being a doctor because I felt so incompetent in the lab. And then gradually started getting hooked on research and never looked back. And I've continued to sort of predominantly do research with a little bit of clinical work on the side ever since.
And when did HBV come into the scene?
So my PhD was quite fundamental immunology. It was very interesting, but I wanted to get it back to something which was more relevant to the patients I was used to seeing. And I had been working with HIV and hepatitis B patients for many years, and actually went to a big international immunology conference and heard Frank Cesari presenting some of their early work that Luca Guidotti had done, showing that T cells had the capacity to fight against viruses without killing infected cells. So the demonstration that CD8 T cells could clear an infection through cytokines and preserve the integrity of the organ was a very inspiring lecture for me. And so I came back and started reading up about hepatitis B, and actually read a lot of work by Antonio Bertoletti on hepatitis B, and then discovered that he was moving to my university. So it just seemed like the stars had aligned for me to work into hepatitis B, and from then on I was working hard to get funding to move into hepatitis B. Actually continued working on HIV first for a bit, and then gradually got pulled more and more into hepatitis B.
John, what's your story?
Oh, well, I've been a nerd since I can't remember when I wasn't one. They were referring to me as professor when I was in 4th grade, and well, now I actually am one. So that's just kind of foreshadowing. I can't ever remember not wanting to be a scientist. It's what I wanted to be since literally I can remember. And I'm having a grand old time doing it. This is just plain fun.
How did HBV happen?
Oh, that's a good story. Well, I was at a symposium being run by my PhD mentor at Penn State, and we discovered the joys of a expense account from a friend who took us out to a bar the night before. And I had a blazing hangover and was sitting in the back row of the lecture hall, trying not to let the flickering lights from the slides make my headache any worse. And all of a sudden, an HBV talk came up, and they displayed a diagram, that standard diagram of the funny-looking HBV genome. And I sat up, bolt upright in my chair, hangover be damned, and said, why on earth would a self-respecting virus do that? And to this day, I still have that question, is why is this virus doing that, and how does it do it? And it's cool.
A malicious little birdie told me that you have an unusual hobby. Explosives?
Yes, I do occasionally explode things. So my wife and I put on a fireworks show on the 4th of July every year.
So very quickly, I've been impressed with the fact that this meeting, by the way, there are 360 attendees, and actually a quick comment. I am impressed with not only the science and the weirdness of this virus, but a seeming dedication from the group to do something good. And I'm hoping that this condensation of all of this into an hour will allow us to broadcast it out to literally tens of thousands of listeners globally from all walks of life, and advertise your mission and recruit more to the fold. So that said, the HBV Foundation coordinates this. Tell us in a few words about the HBV Foundation, John.
Yeah, the Hepatitis B Foundation is the largest patient-focused advocacy group in the world. It is centered out of Doylestown, Pennsylvania, and it is an absolutely superb organization. It helps us here with the science by helping to put on this meeting, but it also reaches out and helps a tremendous number of the members of the affected HBV community through education, lobbying, and just linkage to care. It's a wonderful organization. Those of you who may be interested, I really urge you to go check their website. It's the Hepatitis B Foundation. Just a quick Google, you'll find it.
And we'll put a link to the foundation in the show notes.
So speaking of foundations, nonprofits, Microbe TV, the production company for all these podcasts, is a nonprofit, a 501c3. And we'd love to have your support. If you enjoy these programs, go to microbe.tv/contribute. It's our goal today to give our listeners an overview of HBV, both the virus and the disease. But I want to start by asking each of you to give us a summary, a couple-minute summary of the current focus of your laboratory. So let's start with you, Mala.
So my focus is very much on the immune side. It's a fascinating virus because the body has the natural capacity to maintain immune control in the vast majority of adults who get infected. And so we know that it maintains very strong and effective immune control lifelong, unless people are receiving immunosuppressants. And trying to copy that immune control in people who have chronic infection, and trying to discover how to harness the immune response for immunotherapy has been our long-standing goal. And we're very interested in doing that through understanding more about the liver as a immune organ. So getting a lot of patient samples from the liver tissue to study the local immunity, which is heavily influenced by the very interesting immune environment that you find in this organ, where immunity is very effectively shut down and inhibited by many different cellular pathways. And so our main interest is trying to understand more about that crosstalk between the different immune cells in the liver environment and how that can be harnessed to convert a chronic infection into an immune-controlled infection.
John?
Yeah, my lab has two focuses. They're both on viral replication. For those of you less familiar with HBV, the virus replicates by reverse transcription. And I've spent about 30 years studying the reverse transcriptase, trying to do some biochemistry on a very difficult system. And over the last, say, decade or so, my lab has become very active in drug discovery, and we're actively developing drugs targeting one of the two enzymatic active sites that the viral polymerase has, the ribonuclease H. The other enzymatic activity, the DNA polymerase active site of the RT, is the actual target of the frontline drugs that are used right now.
All right, let's first talk about the disease. I'm going to ask Mala to give us an overview of the different kinds of disease caused by the virus. But John, I'm sure you'd like to jump in as well. So Mala.
Well, it can be broadly divided into acute and chronic. So acute infection actually is most often totally asymptomatic, but it can present with an acute viral illness, with jaundice, liver pain, and vast majority of those cases will then not completely clear the infection, control the infection, I should say, because we know that there's residual traces of the virus following an acute infection, which are then maintained under lifelong immune control. Instead, in chronic infection, which is far more likely to develop if you're infected either at birth or in the first year of life, you can go through different phases of infection. It's an incredibly complicated disease pattern, actually, broadly divided into 4 phases according to the level of the viral replication and the amount of inflammation going on in the liver. But the complications of this chronic viral infection are a necroinflammation of the liver, driving progressive fibrosis, leading potentially to cirrhosis and hepatocellular carcinoma. And hepatocellular carcinoma, which is the commonest type of liver cancer, is the 3rd commonest cause of cancer-related deaths worldwide, so huge problem.
What fraction of chronic infections would end up in HCC?
Gosh, about probably 10%,
I think it's a little bit less than that if you take a look at the entire population. But again, I don't really know. And again, part of the problem is that so many people are chronically infected in resource-limited settings where the statistics that are the basic epidemiology is very lacking. So, that's a very simple question. And the answer is, it's 5-10%-ish, but net lifetime risk, but we really don't know. It's kind of crazy how long we've known about this virus and not know a simple fact like that.
I mean, it's very, very variable, of course. Again, as you say, in sub-Saharan Africa, for example, rates of liver cancer from HBV seem to be much higher, and there's probably cofactors like exposure to aflatoxins that are very important for that. Whereas, in the West, more people are receiving antiviral treatment, which can substantially reduce the risk of liver cancer resulting from HBV.
And how's it transmitted?
So the major route worldwide is mother to baby, which is why it's so important that women are screened in pregnancy if they haven't been tested previously, because ideally all infants should be receiving the birth dose vaccine for hepatitis B. But actually, if mothers are very high-level carriers, they should also be given antivirals in the last trimester of pregnancy to further reduce the risk of transmission to babies. But then there's major other routes of transmission, sexual transmission, contaminated needles, those are the major routes. Yeah.
So the mother to baby, is that during birth, or is it is there fetal transmission?
It can be either, but interestingly, the vaccine will still work if given straight after birth. Yeah.
Tell us about prevalence. Where are the most cases in the world, and how many total cases, say, each year?
Well, the total burden of chronic HBV in the world, again, the numbers are mushy because of where many of these people are located, is ranging between, say, 270 and 300 million people, depending upon the most recent estimate. So it's an enormous number of people. There's, of course, an enormous number of people who have been infected and have had an acute infection. I think the numbers I've seen on that are there's about 2 billion people living right now who have had at least encountered HBV. The largest number of them are located in Southeast Asia, China, Vietnam, India, Indonesia, those areas. But the highest rates of infection are in general in sub-Saharan Africa, where endemicity rates can in some places get 15 to 20% in those areas. The only reason they don't dominate the demographics is that's a less populated region of the world. But the virus is found worldwide. It probably co-evolved with human beings and has spread around the world. So it's everywhere. It's just that the frequency varies.
This is a bit of a squishy question, but I'm interested in your impressions of the sort of global public health impact of the virus, and in particular the chronic infection. You had patients in here talking on Wednesday about what the experience is of being chronically infected and a carrier.
Yeah, I mean, one of the things they were emphasizing is the huge stigma and taboo that's associated with this infection, which causes, a massive psychological burden in addition to the physical risks of long-term complications. And in many countries, you can have substantial blocks to movement and employment if you're found to be S antigen positive still.
So education and therapies can help with that, right?
Yeah, that's very definite. One of the stories that I've heard that was most poignant to me is because there's so much misinformation out there about the virus, but I've heard twice now stories of mothers who have passed the virus unknowingly to their own children, and how devastating it was to them. I mean, you have a new baby, it's your everything. It's your entire focus of life, and then to realize that through no fault of your own at all, you gave this beautiful child a dangerous infection. The guilt level that these women were expressing was just devastating. It was really sad.
I have to say that it is a target of misinformation. Someone who will not be named has recently complained about immunizing children, babies, against a sexually transmitted disease, which completely misunderstands what you've just told us. And so it's really hard to fight that kind of information. Maybe we'll talk about that later when we talk about vaccines. All right, let's move on to the virus particle. John, can you give us a picture of what the HBV virus particle looks like?
Yeah, sure. We'll start from the outside. It's an enveloped protein. It's got surface glycoproteins encoded by the virus that stick out on it. They are glycosylated a little bit, so they've got some sugars on them. And then there is an envelope layer below that, but it's a very unusual envelope in that it isn't a complete envelope. It's more like caulking around the base of the individual surface glycoproteins coming through. There's kind of patches of lipid in there. And then there is a intraviral area that is comprised of one of the surface glycoproteins that acts kind of like a matrix protein that helps the interactions with the capsid. Then you have an icosahedral capsid formed of a single protein called the capsid protein or the core protein. They're both words are used. And interior of that, you have a partially double-stranded DNA in which one strand is slightly over length and has a protein at its 5' end. And then the other strand is on average about half the length of the genome. It goes over the gap in the DNA and has a short RNA oligo at its 5' end. So it's a very strange looking genome.
In addition to the infectious particles, there are also a number of non-infectious particles that are made in high quantity, right?
Yeah. Typically maybe 1,000 to 10,000 time fold in excess over that are excuse me, the subviral particles. They come as either spheres or as tubes of varying length, and those are comprised of lipid and the surface glycoproteins on them. And they are quite important in interaction with the immune system. They act as a beautiful sink for antibodies that are coming up against the virus. And Mala will know more about what it actually does to the immune system, because I'm not a very good immunologist.
Okay, and the replication cycle. It's been described to me as a retrovirus with a bad sense of timing.
That is a very good description of it, actually. Yeah, the reason that and Vincent was kind of teasing us about being a DNA virus. We're really a schizophrenic virus, is what we are, because the difference between, as to why HBV has DNA in its virion and the retroviruses have RNA in their virion is merely a timing of when reverse transcription occurs relative to secretion of virion particles. It really is a minor difference. So the replication cycle, the virus comes in, it binds to the cell through a receptor that was identified by this year's recipient of the Distinguished Award in Hepatitis B research, Wenhui Li. It's called NTCP. It enters the cell and the viral genome is released, enters into the nucleus, is repaired from this funny looking partially double-stranded DNA into a better behaved covalently closed circular molecule. It's a little teeny episome that gets chromatinized. It is the transcription template for all the viral transcripts. Viral transcripts go out in the cytoplasm. They make the various proteins. Then the viral polymerase binds to one of the classes of the RNAs called the pregenomic RNA. That triggers polymerization of the capsid around the genome. Then reverse transcription occurs within the cytoplasm. The nascent virion particle then will interact with surface glycoproteins in intracellular membranes, bud into intracellular vesicles, and then be secreted non-cytolytically out of the cell. So that's the real quick view of the replication cycle. There's two other little steps on there that need to be pointed out because they're quite important. When the nascent virion particles have finished reverse transcription, they have two choices. They can either do what I said about being secreted as virions, or they can be reshunted back into the nucleus in a process that's sometimes a misnomer called recycling, and dump the DNA back into the nucleus to maintain the CCC DNA. And then sometimes when the viral reverse transcription has a mistake at one of its steps, it'll produce a duplex linear DNA rather than the partially double-stranded circular DNA. And then if that gets into the nucleus with pretty high efficiency, those duplex linear molecules can integrate into the genome where they don't contribute to viral replication, but they can continue to produce the viral surface glycoproteins just due to a quirk as to how the nucleic acid is structured as it goes in.
I want to emphasize something you said, just like you tossed it off at the end there. You said it exits cells without cytopathic effects. So is that correct? This virus does not kill the cells that it infects?
That is correct. Yeah, it turns it into a virus production factory. And, the outside world is really scary to a virus. Inside a cell is nice and warm and nurturing. And so why should it kill a cell? It did all that work to infect. And so it turns it into a virus infection factory, and it will continue to produce virus for the lifetime of the cell. It does shorten the lifespan of the hepatocyte, maybe cuts the lifespan in half, but that's still many weeks to a month. So
and during that time, is the hepatocyte doing its normal hepatocyte thing or not?
Is particularly early in infection. There's relatively little direct pathology. Later in the infection, when the inflammation has been building up and there's been chronic damage building up, then of course you do get substantial dysfunction of the cells. But early on, it's quite benign.
And one thing that impresses me, if I understand this correctly, a very small infectious dose can infect a large fraction of the liver. Can you comment on that?
Yeah, the best studies I've ever seen were done by Alison Gilbert many years ago, and she found that for a related hepadnavirus, it's a great model for HBV called duck hepatitis B virus, a single infectious particle is capable of infecting a newly hatched duckling, and then that will roar through the liver, and eventually every single hepatocyte will become infected.
So is it correct that only hepatocytes are infected? And if so, what's the basis for that?
Okay, so there's going to be some people who are angry with me when I say this, but let's rock and roll. The short answer is the vast majority of the infection is in hepatocytes. Some people will say it is exclusively in hepatocytes. There is some evidence for extrahepatic infection in lymphocytes. It's just very difficult to have that be for sure because the level is so very low. But there is evidence, particularly from the woodchuck model, that there is some infection into the lymphocytes that can, under some circumstances, contribute to transmission, but that is really likely to be a very minor component of it. If you just do mass of cells infected, the vast, vast majority of them are hepatocytes, yes.
So what is the role of integration in hepatocellular carcinoma?
That's actually pretty complicated because when the DNA goes in, it is largely non-specific as to where it goes in the cellular genome. It is relatively specific as to where the breakpoints in the circular viral DNA are, but relative to the cellular genome, there's very little specificity to that. And so it goes in there and you're basically having a random mutagenesis going on. And so the virus comes in and it can, of course, disrupt tumor suppressors and things like that. And there are tumors where that's known. It also brings in 2 enhancers and 4 strong hepatospecific promoters. So you end up with promoter insertion mutagenesis. There is also cases where one of the viral proteins is called the X protein. It is a regulatory protein, and it is functionally a tumor promoter, so it is not a overt oncogene, but it does reduce the barrier to cancer through a number of reasons, some of which are known and some of which are not. And then occasionally when you get more severe rearrangements of the viral genome, you will get truncated surface glycoproteins that have some oncogenic behavior. You can also get, because you'll get multiple integrations in a cell, that will promote homologous recombination leading to chromosomal translocations. But the biggest thing it's doing is helping to produce viral proteins because the surface glycoproteins are being produced. And here's where Mala starts talking, and that turns into a stimulus for chronic inflammation, which is also known to be one of the major drivers of cancer.
So is the integration a frequent event? Like most cells that are infected, do they get integrated?
Oh, Thomas, too, should be sitting here next to me right now. Most tumors have multiple infections. Most cells that are infected do not necessarily get an infection in there, but if you count the number of cells that are in the liver, the number of integrations is actually really quite high. The burden can be very high.
And it's actually occurring earlier on in the course of infection than we initially.
Yeah, and it happens really early. Thomas, keep nodding. Did I say that right? Okay, yeah. Thomas, who knows all about this, gives me the thumbs up, so I didn't mess that one up.
All right, let's turn to immunology, Mala. I'd like to do it according to acute versus chronic, if that's possible. So tell us the immunology of an acute infection.
Well, one of the interesting things about the first phase of acute infection is how slow the onset of replication is compared to many viruses, and the relative lack of triggering of innate immune mechanisms, particularly type 1 interferon, which is obviously the normal a first-line defense against viruses is very poorly induced by this virus, and that's why it's been nicknamed a stealth virus, because it can seem to creep in without alerting those first-line defenses. However, there is some indication of other aspects of innate immunity being activated, like NK cells show early signs of activation. And then the resolution of infection has been attributed to adaptive immunity, so primarily a coordinated CD4 and CD8 T cell response, with, as I mentioned earlier, not just the killing of infected cells, but also the clearing of infection through the cytokines that those T cells can produce is critical for clearing infection. We know much less about the sort of immune mechanisms that are driving the development of chronic infection, because it's very difficult to study sort of the onset of chronic infection. What we know is that once you have established chronic infection, you have a profound dysfunction of actually all aspects of the immune response. So NK cells, CD4, CD8, and B cells all show really marked dysfunction. The numbers are depleted and the effector functions are really impaired. And this is thought to be a combination of the years of high antigenic stimulation, as we've heard, particularly the secretion of subviral particles with huge amounts of circulating S antigen, in addition to large amounts of virions bombarding the immune system for decades. But also the what we call tolerogenic liver environment, where the liver has these complicated mechanisms for shutting down immune responses. Because the liver receives blood supply from the gut, the liver is an organ which is bombarded with foreign antigens all the time, food antigens and microbial antigens. And so it's evolved numerous mechanisms for limiting immune responses. And one of the interesting things about hepatitis B virus is that it can be presented to the immune system by the cells of the liver, the hepatocytes themselves, rather than the classical antigen-presenting cells that are good at triggering an effective immune response. The hepatocytes can present to the T cells, but they induce this poorly functional T cell response.
In an acute infection, is there a time when there's no more closed circular DNA? Is it eventually cleared, or is there always some lingering?
No, that's not the case. It's actually, what we think sort of in the vast majority of cases, there's persistent traces of cccDNA DNA as well as integrated DNA following resolution of acute infection. And that's very clinically evident because people who've resolved hepatitis B have developed anti-S antibodies, if they're then immunosuppressed, given chemotherapy for cancer or have an organ transplant, they're at risk of reactivating hepatitis B. And interestingly, actually, even if they're just given a drug which depletes their B cells, they can reactivate hepatitis B. So that tells us that the humoral immunity as well is important in maintaining that long-term immune control.
So my understanding is that the probability of developing a chronic infection is much higher if you're infected in infancy. Why is that?
Yeah, the concept has been thought to be related to the sort of tolerance that you get from mother to child transmission, potentially thymic tolerance being relevant there. But also the exposure to a very immature immune system in a baby or infant is likely to be relevant as well. And we heard yesterday at the conference about the secretory form of core antigen, the E antigen, that we haven't talked much about yesterday, but that, if that's transmitted across the placenta, has been shown to be able to specifically shut down or tolerize the infant immune response to hepatitis B.
So the fact that circulating non-infectious particles are binding antibody, as John mentioned, this implies an important role for antibodies as well in resolution, perhaps?
Yeah, I mean, as I said, the fact that when you deplete memory B cells with anti-CD20 drugs, so drugs like rituximab that are used for autoimmune conditions and for lymphomas, you get reactivation of hepatitis B, tells us that not only are antibodies important for prevention, of course, that's the basis of the vaccine, but they're actually also important for maintaining long-term immune control following infection.
So from your immunologist point of view now, could you comment on the mechanisms of oncogenesis?
So I mean, the chronic disease that you get in hepatitis B, because we've already heard it's a non-cytopathic virus, so the virus itself can replicate at incredibly high levels without doing any damage to the hepatocytes, and the immune response to the virus is what's driving the disease, both the inflammation, the fibrosis, and a very important role in oncogenesis.
And so how does the integration play into that?
Well, the two come together. So you have the oncogenic effect of integration in tandem with this. Most cases of HCC will arise on a background of chronic hepatitis and cirrhosis. Not all of them, but the vast majority.
I'm curious, in a chronic infection, you see a lot of S antigen circulating, right? Do you have any feel for what fraction of that comes from integrated genomes and what fraction is from closed circular DNA?
I mean, that depends on the stage of infection. So once you reach the later stages of infection, which are characterized by being E antigen negative, the vast majority of S antigen is actually coming from integrated DNA. We've only really realized that in recent years.
This is a crazy virus.
That's true, but it's also it's scientifically fascinating, but devastating to people.
John, do you have to be crazy to work on it?
Looking out at my friends here, I would say yes. I mean, yeah. Oh, one of the other things that makes it crazy to work on HBV is we're really fairly constrained in the models that we have. It was only relatively recently that we would even get a cell line that you could carry in the lab easily that we could actually infect. I mean, that's just crazy. I mean, the animal models, we've got a number of them that isolate different aspects of the viral infection, but it's very difficult, nearly impossible with HBV itself to have a true infection in an immunocompetent animal. So a lot of the advances that occur in HBV are really tightly coupled to the technical advances that we generate in the systems we're working with.
Yeah, in fact, famously, duck hepatitis virus played a big role in elucidating a lot of the molecular biology, right?
Yeah.
So do ducks get hepatitis B in the wild?
No. That's one of the reasons why I no longer study it, because talking the NIH into studying a virus that doesn't even cause disease in its natural host got a little hard after a while. So, yeah. But no, it's a superb model for the molecular biology. The entire reverse transcription pathway was worked out in DHBV and then confirmed in HBV. So it is a superb model.
I forget whether you mentioned it's at 2.5-ish kb. It's like the smallest.
Well, it's 3.2 kb.
3.2, sorry.
Yeah, it's a tiny virus.
Right.
But it's also the most genetically dense piece of information I know, because every single nucleotide in the virus codes for more than one protein at a time in different open reading frames. You've got, again, the 4 promoters, 2 enhancers, all the cis-acting signals related to reverse transcription. There's a number of cis-acting signals related to RNA processing, and it's just a crazy dense piece of information.
Mala, I wanted to just ask a few more immunology questions. First of all, you mentioned that there's not an innate response to infection. So does that mean that people with defects in interferon, whether it's genetic or autoantibodies, they don't have more severe Hep B?
That's very interesting, actually. A colleague of mine is just looking into the prevalence of the anti-interferon antibodies and what impact they have on chronic hepatitis B. So we don't know much about that yet. And I don't want to say that there's no interferon response. This is quite a controversial area. And probably during chronic infection, when there's lots of inflammation, you can start to get a bit of innate immune responses. But it seems like there's predominantly a lack of effective induction rather than a very active inhibition of type 1 interferon, which you see in, for example, hepatitis C.
Okay, one more from me. You said CD8s and CD4 cells were important, antibodies, CTLs, of course. Any other cell types like NK cells, any FC-mediated functions are known to be important for protection or clearance?
Yeah, I mean, NKs potentially have the capacity to play a major antiviral role because they can recognize the lack of class 1 on the infected hepatocytes and upregulation of stress molecules. But work from our lab and others has shown that they're also important in regulating other aspects of the immune response, particularly in down-regulating the T cell response. So trying to harness NK immunity comes with challenges because you would want to try and promote their antiviral capacity whilst preventing them from then limiting the T cell response.
Okay, the last section, let's talk about therapies. And Rich, I'll hand it to you.
Yeah, just to start off with, my understanding is we have therapies in several different classes targeting different parts of the virus and the replication cycle. John, if you could comment on replication inhibitors.
Okay, so there are the frontline drugs right now are replication inhibitors. They're nucleoside or nucleotide analogs. They're very good drugs. They suppress viral replication by typically 5 to 7 logs in the large majority of patients, but they're not curative. And so there's a huge push going on right now to develop curative therapies. So the ones that are out there right now that are targeting replication, the leading classes of pure replication inhibitors would be, for example, the capsid assembly modifiers, which are really, really interesting molecules that act by actually accelerating the assembly of the capsid, and that sends it down a couple of different non-productive pathways. It's truly a fascinating mechanism there. There's many of those that are in clinical trials, some of them doing quite well. There are other forms of replication inhibitors. For example, my lab is working with ribonuclease H inhibitors. That is a pure replication inhibitor. There is room for those in therapy in my mind because the current drugs cannot suppress viral replication enough, and we do have to hammer on it harder. So that's great. Then the other classes that are really gaining a lot of traction right now are siRNAs and antisense oligos that are involved in binding to and messing with the function of the viral RNAs. That has the dual effect of suppressing antigenemia, which is handy to help reduce the inflammation and some of the other damage that's going on there. But of course, you're also destroying the viral pregenomic RNA, so it's a direct replication component inhibitor, and reducing the accumulation of the proteins involved in reverse transcription. And so those are probably the leading classes right now. But there's a huge number. Basically, if there's any aspect in the virus, there's somebody trying to hit it with a hammer right now.
So what would currently be the sort of standard therapy?
Well, the current standard therapy is monotherapy with either an entecavir or one of 2 prodrug forms of tenofovir, and they're both nucleoside analogs. They're chain terminators. Stop DNA replication by simply terminating it.
And so this would be, is this applicable to both acute and a chronic infection?
They don't really treat acute infection, and I'm not a physician, and so I can't really say why that acute infection is not targeted. So they're used almost exclusively in chronic infection. For historical reasons, I think, and again, I'm probably going to get a lot of people angry with me on this one, they have been used only after overt illness starts developing. There's a very long lag period for most people who are infected with HBV before there's overt disease that is detectable in the individuals where they start feeling bad and stuff. And so the guidelines for treatment with these nuke analogs have always focused for you got to have some evidence of liver damage and a high level of DNA replication, are really the core components of a complex algorithm. And a lot of us are thinking that is occurring, treating is occurring much too late, because the data that's coming out of the people that are sitting in this room is indicating that the chronic inflammation induced by the virus, even when it's subclinical and you're not feeling it, is causing damage to accumulate in the liver. And the steady accumulation of integrations is causing damage in the liver. And so there's a large group of us that are working right now to push for broadening of the treatment guidelines. And some progress on that is going. The Chinese guidelines have recently been broadened quite nicely, and the WHO has just revised their guidelines, again, broadening them. And so that's a very active area, but in short, it's nucleoside analog treatment of chronically infected people with overt disease is where it's going now.
And just to say, it's actually, I mean, the major issues that most people who have this infection are not even being diagnosed. We know about less than 10% of the cases that are out there. And then most people who are diagnosed are not receiving treatment. So in sub-Saharan Africa, I think only 2% of the infections that exist are diagnosed, and obviously most are not receiving treatment. We are, even in cases where treatment's available, withholding it, and as John said, until quite late stages of the disease. So there's a massive issue with underdiagnosis and undertreatment of this infection.
So am I understanding that if you could treat as soon as possible after exposure, you might be able to prevent persistence of cccDNA?
I doubt that. I really doubt that. You're really going to need the immune system to dig the cccDNA out or put a lid on it so that it doesn't cause long-term problems. The argument I was making about earlier treatment is to reduce the rate and frequency of severe disease progression. That's what the goal of earlier treatment that I was advocating for is.
Are there immune-based therapies?
Well, of course, interferon is partially immune-based, and that was traditionally one of the mainstays of treatment. It's now largely been replaced by the much better tolerated nucleoside analogues. Entecavir and tenofovir are the mainstays of treatment. But interferon alpha treatment has been used for years for hepatitis B. It works both through direct antiviral and immunomodulatory mechanisms. And it's being sort of seeing a bit of a revival in some of the new functional cure trials being used again in combination with some of the newer antivirals. And then, of course, we're working hard in the field to try and develop more targeted immune therapies, which work in a more specific way than interferon. And one of the mainstays of potential immunotherapeutic strategies is to try and induce new immune responses, because the existing ones are so exhausted. So a potent therapeutic vaccine, if given in the setting where the viral antigen has been substantially reduced by some of the newer drugs, potentially also with other immunomodulatory approaches to rescue dysfunctional responses, is a sort of exciting prospect for the future.
So I want to be sure we're clear on, or everybody's clear on this concept of a functional cure, okay? So the implication, and I think the reality is that means that the virus and its bits are not gone. So if you could clarify that and comment on what happens if therapy is withdrawn.
Yeah, so a sterilizing cure would be actual elimination of the virus, and that, as John's already said, is probably unrealistic in the near to mid future. But a functional cure just defines loss of surface antigen and HBV DNA in the periphery with 6 months off therapy. So it's the concept that you, instead of having to maintain patients on lifelong therapy, which is the standard treatment at the moment, you would be able to withdraw therapy and not see a recrudescence of detectable viremia and S antigen in the blood.
And that would largely mimic the natural clearance of an acute infection, reaching a state where there's still a little bit of the viral DNA around. Some of it is probably in a functional manner, but the immune system's got it under glorious control.
So would that mean that a person presents clinically as if they did not have anything going on according to the normal tests that one would do?
Well, you would probably at that point you would certainly still have some of the antibody markers that would come up, but you wouldn't have any of the viral markers, with the exception, well, again, the part of the problem in defining a functional cure right now is that the functional cure requires removal of detectable surface glycoproteins from the serum. But we know that in late stage patients, the large proportion of that is coming from integrated non-replication competent versions of it. So we have many people who have really actually achieved a functional cure but don't meet the current definition because we don't have decent biomarkers. That's really actually a huge problem.
I think we need to discuss the vaccines that are available. One of you, can you tell us what they are and what they do?
Well, we've got preventative and therapeutic vaccines. So the preventative vaccine is based on the surface antigen, so predominantly inducing an antibody response to protect against infection and, should, as I said earlier, be given to all newborns, but not all countries have implemented that yet. And even the ones that in theory have implemented it are having great difficulty in rolling it out effectively. I mean, even the UK has only just started in recent years to give it to all newborns. It has to be given usually as 3 doses, which is quite a problem with compliance. And then 5 to 10% of people are non-responders. But it's a very safe and effective vaccine, and it really needs to be given to a lot more people. So, getting this vaccine out there and getting people to understand the importance of it is really, really important.
Maybe you said this, but the vaccine it's a subunit vaccine consisting of recombinant made S protein, right?
That's right, yeah.
And therapeutic vaccines?
So therapeutic vaccine is a concept that once you have chronic infection, you can try to, rather than giving the standard preventative vaccine, you give a more immunogenic vaccine consisting of usually all the major HBV proteins to try to stimulate new T and B cells to replace the dysfunctional ones that are failing to control infection. There have been many trials of therapeutic vaccines over the years in chronic hepatitis B, which have not yet been successful, but they probably haven't been given to the right patients with the right immunogens and in the right combinations. And now we have the capacity to really tailor both the vaccines and the patients who receive them so that we can try to optimize responses by first reducing viremia and viral antigens, then giving a more immunogenic vaccine and possibly combining it. So the checkpoint inhibitor drugs that were best known for use in cancer, things like PD-1 blockade, are being also given in some trials in combination with these therapeutic vaccines, and they're improving responses to the vaccines.
All right, we have one last question for each of you, and I would like to ask Mala, what would you have done if you had not become a scientist?
Well, I actually wanted to become a writer, and in fact, I do a lot of writing in science, which you maybe wouldn't expect when you start out in science, that writing is a big part of, and communicating is a big part of science, which I love. But I would like to, at one point, write something more creative than scientific writing. So I hope to get round to that one day.
John?
Well, I always make the joke that if there were jobs like for professors, I'd be living in a cardboard box on the street because I'm really not suited for anything else. But if I were suddenly to become independently wealthy, which is highly unlikely, I think I would just go down to my woodworking shop and continue making furniture and wooden bowls and sawdust and have a grand old time.
All right. Thank you very much. That is TWiV from HBV 2024. You can find the episode. It'll be released in some few weeks at microbe.tv/TWiV. Questions, comments, TWiV@microbe.tv. And as I said earlier, if you enjoy these science programs, we would love your support to continue them. We don't do advertising. We exist on the donations of our supporters. We have several ways you can contribute. Microbe.tv/contribute. And in the U.S., your contributions are U.S. tax deductible. Our guests today from University College London, Mala Maini. Thank you so much for joining us. From St. Louis University, John Tavis. Thank you so much. Rich Condit is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
It is always a good time, and it's been a real privilege to be with everybody.
I'm Vincent Racaniello. You can find me at microbe.tv. I would like to thank the organizers of HBV 2024 for having TWiV, Alex Plus, and Matteo Iannacone. I get to say an Italian name. Oh, boy. I'd also like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Thank you.